good weekend. Stayed warm. This was a very, very cold weekend. Oh, good afternoon. All right, while we're waiting for everybody to get in, if you could go to your Canvas page and look at Punnett squares, monohybrid hybrid crosses. Okay, so I'm only missing a couple of people, but again, while we're waiting for them to come in, everybody be Canvas, okay, and looking at this unit on monohybrid and dihybrid crosses. We have been talking about genetics, and we are looking at, last time we talked about um, Mendel and his work with pea plants. And so what we're going to do today is practice uh, looking at what he was doing. So we're going to give it a few minutes to let a few more people try to come on. I appreciate you all being on time. All right, and then there's going to be a worksheet that you're going to upload for me today. So give this a read, and then we'll do this together. All right, I'm going to stop sharing for a minute, do attendance, and we'll just go forward with who's here. Everybody else is late. Oh, as soon as I say that, somebody comes. Oh, good afternoon, Andrea. We are in Canvas, and we are in the unit on Punnett squares and monohybrid cross. I'm going to do attendance, and then we'll get started. Still missing a few folks, but we're going to get started anyway. Okay. Okay, Michaela is not here. Other than that, everybody's here. Okay. The last class told me that they remembered how to do Punnett squares from middle school. How about y'all? Do y'all remember how to do how to do Punnett squares? I got one thumbs up. How about the rest of y'all? Y'all, everybody just give me a thumbs up. Remember. Okay, so then I'm gonna just review this real quick and then let you get to the activity. So again, read through this. What you need to remember with Punnett squares is that it is a probability. When we did meiosis and mitosis, um, for meiosis, we said that that happens in the sex cells, and that determines most all of your maleness and your femaleness characteristics. However, those during meiosis, the cells divide and create four cells. So if you remember with the at the end of that, we had four new cells that had the haploids number of chromosomes of the original parent. So when we do Punnett squares, we're going to be looking at that. We're going to take the parent and then we're going to divide their chromosomes into boxes, and then we're going to look at the probability of what kind of offspring we can produce. And we do it with a box, four boxes, and the box represents 100%. So each box represents 25% possibility that you're going to get one type of offspring or another. And so we're going to look at this in terms of genotype and phenotype. So we're going to put the genes in the boxes, and then we're going to look at how do we describe that genotype. And then we're going to look at, based on that genotype, what does this offspring look like? So that's what we do with Punnett squares. Okay. All right. So again, with your inherited chromosomes, you get X and Y from dad, you get XX from mom. And so if that's all they have to offer, then the possibility is that during meiosis, dad can give an X or he can give a Y. And if he gives a Y, and mom can give both an X and an X because that's all she's got to give. But if he gives an X to her Y, I mean, if he gives a Y to her X, then you're going to have a boy. If he gives an X to her X, you end up with girls. So that's how we call that segregation. So we had some vocabulary words, law of segregation, in that the chromosomes spread out and they go in different boxes and they are fertilized in different cells. So they're not all just being donated at the same time, which is why we get the variety of things that we get. And down here at the bottom of that box, it talks about probability. So again, we have that word probability, and we can express it as a fraction, or we can express it as a percentage. Most of the time, we're going to express it as a percentage or a ratio. Okay. All right. So you all said you know what to do with a Punnett square. This is a Punnett square, and one parent's genes are put across the top. Or the trait that we're looking at, we're going to take these one trait at a time, and that would be a monohybrid cross. We're going to take them one at a time, and the other parent's genes will go down the side. And then we will take one from each parent and place it in the box until we fill all the boxes. And then the same thing happened when he was looking at the pea pods and characteristics of color and shrinkage and all that sort of thing. So I'm going to show you the other characteristic on here. Okay, I'm gonna come back to that in a minute. Actually, I have both of those on my Canvas screen. So let's switch to that. And I'm not sharing, am I? I thought I was sharing. Okay. Okay. All righty. Sorry about that. what I was doing when I thought I was sharing, I was looking at this box, taking you through this box in the middle of the page. And so we are on Punnett squares, and so that's what we're going to look at. So I'm going to take you to a sample. So we have two words that are in our page today: monohybrid cross and dihybrid cross. I'll tell you, this unit has a lot of vocabulary, and you just have to be able to recognize what we're doing. So mono means that we only are looking at one trait. So in this case, we are looking at the trait for the color of the seed, okay? And it's hybrid, so there's two different colors. So we have a yellow color and we have a green color, but we're only looking at one trait. That's why it's mono. Okay. All right. So again, we start with the parents. And again, this is a review so that you can refresh your mind, because you said you did this and you remember how to do it. But I'm going to walk you through it anyway. So in our parent generation, our parents have two chromosomes, and they're alleles because the alleles have the same trait on them. Okay, so this one, the first one, has yellow, and there are capital letters, so that tells us that yellow is the dominant trait for the shape of the seed, the round of the seed. We got a lot of characteristics that we're going to look at, but the seed is going to be yellow. And if it's dominant, it's dominant if it's yellow, and recessive if it's green, because we express that with two lowercase y's. So that's our preliminary. We know we have one seed that is yellow and one seed that is green. And we have their gametes. Notice they use the term gametes. So that's what we were saying when during meiosis and they create the reproductive cells, the gamete from one parent, and because they're both the same, they can only give capital Y gametes. The other one can only give lowercase y gametes. And when they get to the first generation, the only type of offspring you can get with the first generation is capital Y, lowercase y, because that's what is given. There's no other choice. So everybody in the first filial generation is going to be heterozygous, and they're going to be yellow. Notice the box is yellow because yellow is dominant. So this is how we have to read this. We have to be able to say that it is heterozygous because we have two different genes, and it is heterozygous for yellow. And that's our phenotypes, our genotype and phenotype. All right, now with our F1 generation, we're going to take two of those offspring, and because they can give either one of these gametes, the box for the F2 generation will look like this. Across the top of the box, we have a capital Y and a lowercase y. And then on the side of the box, we also have a capital Y and a lowercase y, because we're crossing two members of that F1 generation, and all of them look the same. Okay? Everybody in the F1 generation, and remember that that's a rule. F1 generation is, let me back up. If you cross a pure dominant with a pure recessive, your F1 generation will be 100% heterozygous. Okay? Now we're taking our heterozygous offspring and we're crossing them. And this is what we can get in the F2 generation. We can get 3 boxes, if you see here, that have yellow, but they're not all the same. So there's a couple of ways we can express this. And 1 box that is green. So we can express this if you look down to the bottom. The genotype for those 3 boxes is going to be yellow. But we express the genotype. There's 1 box that has 2 capital Y's, and then 2 boxes that have a capital Y and a lowercase y. So 1 box is homozygous dominant for yellow, and the other 2 boxes are heterozygous for yellow, because they have 2 different genes, but they're still yellow. Okay, so again, we look at it at genotype. There's 1 of the homozygous one, and there's 2 of the heterozygous. But if we're looking at how many yellows we have 3 yellows. Okay. Then we have the green. There's only one, the one with the 2 lowercase y's. We only have one of those, so the genotype ratio for that is 1. And the phenotype ratio is also 1. So again, if we're looking at this as ratios, it's 1 to 2 to 1, or it can be 3 to 1 if we're looking at phenotypes. Okay, we could also call that a percentage. It could be 25% of the homozygous yellow and 50% heterozygous yellow and 25% homozygous green. Okay, so you see the different ways I describe that. That's how you have to be able to describe it. So the worksheet that we're going to do is going to ask you to identify what they're giving you in those terms. All right, so the other term that you have on that page is dihybrid. So there is no chromosome has just one characteristic. Think of a chromosome, I said this before, as a string of pearls, and there are several different traits on that string. Okay, so the chromosome, first chromosome we just looked at the one trait that was on that chromosome. That chromosome can have up to 25, 30 different traits on it. So in this next box, we're only looking at 2 traits. So we're looking at a dihybrid cross. So we're looking at whether it is, and what are they looking at here? They're looking at the shape of the pod. Okay, it doesn't say it there, but that's what they're looking at, the shape of the pod. So the pod can be yellow. Sorry, the pod can be round, and they're taking the letter R for round because round is dominant, or the pod can be wrinkled. So we're looking at, is it yellow or green, and is it round or wrinkled? Okay, yellow or green, round or wrinkled. And they can, and notice how they put both of the characteristics for each parent in the same box. So the parent had the dominant parent that has both capital letters, has both capital Y's and capital R's. Again, we're using R to represent round because round is dominant. Okay, on the other parent has 2 lowercase y's because they're green, but they're also wrinkled, so they're using lowercase r's. Okay, so when we cross those, here again, we had a pure dominant with a pure recessive. F1 generation is 100% heterozygous. So we had a capital Y and a lowercase y, a capital R, and a lowercase r. 100 for the F1 generation is always 100% heterozygous. Now, the good trick comes when we have to do the next generation. So if we take 2 of these individuals and we cross them, then we've got to separate out these traits into pairs. Okay, so we're going to take an R. When we go through meiosis, the chromosome with the Y and the R is going to be on one strand and another one on this one. And the lowercase y is on one and the lowercase at YR and another one with a YR. Okay, so this is what we have at the top. We have one parent, the heterozygous parent, and they had, if you look at it, we still have 2 Y's, 2 capital Y's, and we still have 2 capital R's, but we've segregated them. We separated them out and paired them up. So we have 1 and 1. Okay, everybody following so far? You see where we got for the dihybrid cross, why we separated it out that way? If notes, type in the chat note, say that again. And then on coming down to do the same thing with the other parent because they're both the same. So we're going to put, okay, since they're both the same, we're going to have the same at the top and the same at the bottom. Okay, I mean, coming down the side. And we do the same thing again. We take the letters from the top and move with the side, and it's, you know, easier to go one row at a time. So you're moving that one with everybody at the top, then go to the next row, do everybody with the top until you complete that box. All right, so then reading this box has more things on it because we now have twice as many. We actually have 4 times as many because instead of having 4 results, we now have 16. All right, so when we look at the phenotype of these, we now, if you count all the yellow boxes, we have 9 yellows, but they're not all the same. So we can go back and look at the first box has capital, so that's dominant. So that's the only one that is going to have 2 capital R's. So there's only one of them, 2 capital Y's and 2 capital R's. All the rest are going to be some sort of heterozygous of some sort, but they're still going to be yellow. All right, then we have the green ones. The green ones are all going to have lowercase y's, but notice they can have capital R's. So you have the difference there as well. So you have 3 of those. You have one yellow one that has that happens to be wrinkled because it has 2 lowercase r's, but it's still yellow. And then you have one green one that is both wrinkled and green out of all of those crosses. Okay, so that's what your objective is today, is to complete the worksheet so that you are answering those questions just like that. All right, let me come back over here and tell you where that worksheet is. This one for you is in the module because I transferred it. So let me see. So I'm back in Canvas and I'm going to, all right, there we go. All right, so you should just go click advance to the next space on your module and you should find this. Okay, let me know if it's not there. Or if you go back to the module that we were working on and then just hit next, it should be your next module. Okay, and then when you open it, yay, it opened. Thank you, Kendall. All right. Oh, did I? I did. Okay. And you should have, let me view this as a student to make sure that you have a folder. Okay. All right, so if you start assignment, yours should look like that, and then that way you can upload it when you're done. Okay, so you should have downloaded a worksheet that looks like this. Okay, so each time they will tell you what characteristic is dominant. So in this case, we're looking at the pea pod, and they're telling you that an inflated pea pod is dominant over a constricted pea pod. Okay, so we're going to use, for the sake of uniformity, we're going to use the letter I for inflated. Okay, and so you'll use 2 capital I's for the first one, which is inflated, and 2 lowercase i's for constricted. And then on the other side, they're telling you that yellow is dominant to green. So let's again use the Y as a capital Y and small case y for the green one. Okay, and let's see, is there anything else we need to decide? Purple and white, we were using P for the dominant trait and small capital P and then lowercase p for white. And then on the other side, they have short is recessive, tall. So let's use T for tall and lowercase t for short so that we're all doing the same thing at the same time. All right, So I need you to work on this worksheet and then upload that for me. And when you finish that, I'm not leaving this site. Oh, cancel because I'm in student mode. Okay. When you finish that, go back to the Canvas module and make sure you've done this activity and this one. So just complete the activities on this page and look at before you go. All right, so but right now you're working on this. But I'm saying if you finish that before class ends, you need to go back to Canvas and finish that Canvas page. And definitely ask any questions you might have. So again, we're using I for inflated, and we're using capital I and lowercase I, and then we're using capital Y and lowercase y for the colors. Capital P for purple and lowercase p for white, and capital T for tall and lowercase t for short. All right, and as I said to the other classes, you have class time to do this, so everybody who's present, there's no excuse why this assignment should be late, because if you are present, you should be doing this assignment during class. What I do need to make sure, is to change the due dates on this. So let me do that. Yours is different from the other class. 14th, that's tomorrow. So, all right, so yours will be late after the 15th. Even though I'm saying that you should be able to get this done in class today, on the 15th, it will be late. Okay. Okay, because inflated is capital, is dominant. So we're going to always use the capital letter for the dominant characteristic. Just always remember that. It's always the capital letter for the dominant characteristic. They tell you that inflated is dominant, so we're going to use capital I and then lowercase I for the recessive trait. On the other side, they're telling you that green, I'm sorry, They have green. They have green and yellow. So I said the other way around. That green is dominant, so use G as capitals. Sorry about that. That green is dominant. For this is for the pod case. It's not for the seed itself. For the pod case, green is dominant. So you're going to make capital G and lowercase g for yellow. I apologize. Green is dominant. Always use the dominant capital letter. Down here, purple is dominant. So we want capital P, lowercase p for white. Over here, they tell you that tall is the dominant trait and short is recessive. So you're going to use T for tall and lowercase t for short. Now for these boxes, like for number 2 and number 6, you're actually going to have to write your letters on top of each box and place in the box what would the cross be. And when they say what is the phenotype, in the first case, is it inflated or is it constricted? On the other side, are they yellow or are they green? um, when you do the heterozygous cross, how many of them are going to be inflated? So you can just say 3 or 4 or 2 or 1. For those, you don't have to give me percentages on those, but just count what you see in the box. This is about, are you able to read what you put in the box? Which, again, green is dominant, so be using G. If you have any questions, please ask. All right, Malia, are you still with us? Okay. My late student. All right, I'm going to have to repeat this because Michaela is coming in. Okay. Okay, good afternoon, Michaela. I'm sorry, because I didn't see, because I would just check the Go Guardian, so I just saw you there. Um, we are in Canvas. If you go to Canvas and you go to monohybrid crosses and dihybrid crosses, monohybrid and dihybrid crosses, read over that page and at least as far as what the Punnett square is. No, 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 I'm talking to Michaela. So not Kayla. Michaela. Michaela. Okay, so Michaela just joined, so I'm trying to explain to her what we are doing. Okay, and so if you come down to the bottom and you hit next, we are working on the work, and you download this worksheet. The worksheet should look like this. We're using capital I for inflated as dominant and lowercase I for constricted, and we're using capital G for green and lowercase g for yellow, so that we're all doing the same thing. Okay. The rest of the class told me they remembered how to do Punnett squares from middle school, but if you need some help, let me know. Actually, let me pull this back over as this was the demo that I had up. So I'll leave that up to give you some guidance. All right, now for question number 3, make sure that you read that correctly. For question number 3, we're not taking two individuals. They specifically want you to cross a heterozygous inflated, so that's a capital I and lowercase I, with a homozygous constricted, so that's two lowercase i's. So make sure that your number 3 is like that. And then for number 4, they want you to cross two heterozygous inflated. So that's what we've been doing. But for the second one, for number 3, it's a homozygous constricted. So make sure you follow that. This is why I said vocabulary is important. You have to make sure that you are um, recognizing what letters you're going to put in the box based on what they have described. And they do the same thing on the second page where they're specifically asking you to do a heterozygous purple flower with a homozygous white flowered plant. So make sure you read those carefully. And for number 8, so that we're consistent, let's use A for the axial flower. Since the axial flower is dominant, let's use capital A and a lowercase a for the terminal. Oh, and I forgot to say, when you have the boxes, the three boxes for the up here at the top, you want to say, for example, the first one is going to be homozygous. Um, and I'm telling you the answers. Homozygous. Actually, it's already. No, it didn't come out. I had typed that on mine. Homozygous inflated pod, a heterozygous inflated pod, and a homozygous constricted pod. So that's why there's three lines. So you need to distinguish homozygous and heterozygous. Same thing with um, the colors of the pod. You want to say homozygous green, heterozygous green, and homozygous yellow. So for phenotype. And then make sure you do the same on the next page with the purple and the tall and short, purple and white, tall and short. Okay, and again, you should have enough time to get this completed and uploaded today so that you're not late. Trying to get away from lates. And having done that, go back to the Canvas page and um, finish reading out that page and do the activities on that page. Watch. All right, so we got a few minutes left to class. Again, the objective is to get this completed and uploaded before you log off. So you got about 5 more minutes. 8. On the first page or the second page? For the first page, it's going to be A. We're going to, for axial flowers or dominant, so we're going to use capital A and lowercase a. And again, for the second page, it's going to be tall and short, even though they're saying dwarf. We're going to use capital T and lowercase t. You're welcome. All right, it's almost 1:30. Anybody have any questions? Yeah, I'll be here for a few minutes. I know some of you have another class that you have to log into.